Hello everyone. So we will now hear three presentations from the imagery team. The first one is from Olivier Villemin, and he will teach and explain his subject, myocardial stiffness assessment by ultrasound.
Cool. Good afternoon, everyone. In English, right? Yeah, not sure. Okay, let's do it in English. So, indeed, so three presentations. And the first one will be more like a theoretical presentation on myocardial stiffness and the quantitative assessment by ultrasound. But before talking about ultrasound, let's just to be sure that there is no confusion about myocardial stiffness. I'm saying that because if you ask to a cardiologist what is stiffness, usually the cardiologist will think about the chamber stiffness. And I think it's extremely important to separate the chamber stiffness, which is by definition the structural stiffness, which is a combination between the material stiffness, the myocardial stiffness, plus the chamber characteristics, so basically the mass, the size, and the volume. And one simple way to assess the chamber stiffness is to use the relationship over time, the ratio between the pressure and the volume, which is usually a good way to assess the chamber stiffness. And at the opposite, even if it's linked, there is the myocardial stiffness. And the myocardial stiffness, by definition, is intrinsic of the material. So basically, it's the material stiffness. It's extremely interesting because it means that it's independent of the geometrical characteristic of the pump. The issue that we have in cardiology is that the myocardium itself is a very, very complicated tissue. It's a nonlinear, viscoelastic, anisotropic, active material. And you will see that each adjective is important, and I will try to explain that in my presentation. So, when I was a medical student, it's really like a paradox because I still have in mind my mentors. I was young, and my mentor said to me every time, yeah, myocardial stiffness is really key in cardiac physiology. It's extremely important. You will find tons of papers on the myocardial stiffness in very like big journals in myocardial stiffness really is extremely important, but impossible to assess. It's a weird situation. It's a paradox because we know that myocardial stiffness is one of the key parameters for the cardiac physiology, for the diastolic function, but also for the systolic function, for the contractility. And that is why, pretty recently, some authors said that it's the holy grail in cardiology. So if we could have access to the quantitative assessment of the myocardial stiffness, basically, we could better understand the cardiac physiology. So when I said it's impossible to assess, it's not true anymore, I will say. If you look at the way that we could assess the myocardial stiffness for in vivo assessment, the gold standard, even if the expression is a bit weird, but the gold standard is an invasive assessment. So we use this relationship between the volume and the pressure in order to assess the chamber stiffness and then to deduce the myocardial stiffness. However, as you could imagine, for human application, it's challenging because it's an invasive assessment. So there is some risk linked to this type of exploration. There is two types of imaging technique, which could be extremely interesting for tissue characterization. There's the MRI techniques. I guess that we could have some talks dedicated on elastography by MRI. It's extremely interesting. And the idea under the MRI assessment is almost the same as the ultrasound assessment. You will stimulate the body, you will induce some vibration, and using the reaction after the vibration, you could assess the viscoelastic properties of the tissue. So basically, if you look at the MRI assessment, you have a driver which will induce a vibration, and using this type of stimulation, you could quantify the stiffness by MRI techniques. And naturally, you have also the ultrasound. So when I say stimulation and reaction of the tissue, I think if you just keep this information from my presentation, it will be enough. the way that we could quantify stiffness using imaging technique is linked with the velocity of some events through the tissue. And in our case, the velocity of shear waves or natural waves. And we know that the velocity of this wave are directly linked with the stiffness through some elastic modulus, such as either U modulus, even if it's not perfectly applicable in myocardium, or the shear modulus, when basically if you could quantify the velocity, you could have access to the stiffness. So why my introduction was a bit long is that if you understood that it's extremely difficult to assess myocardial stiffness, and the only way to assess myocardial stiffness is to see this very fast wave, you need an imaging technique able to have a very high frame rate to see these waves. And so, naturally, ultra-fast ultrasound imaging, I already gave some presentation on that, increase a lot the temporal resolution, for ultrasound. And so you could see this wave propagate in the medium, you could track them, and you could assess their velocity. So when I said this wave, there is two types of waves. And to me, I have a bias, but I think it's extremely interesting. You have natural wave. So all of us right now, we have many natural vibration in our heart due to natural events, such as valve closure or contraction. And so many teams try to see this type of vibration, this natural vibration, natural waves, and use these velocities to better understand the viscoelastic property of the myocardium. The other way is to induce these waves. So you could basically use some mechanical characteristic from the ultrasound and induce a push, a tiny stimulation in the myocardium, and then track the reaction of the medium, of the myocardium, and basically see these waves. So in real life, we demonstrated that it's possible. We could induce shear wave propagation on the myocardium on beating heart on human. So just a few examples, just to demonstrate it that we just started very simply, like every time I will say almost in this type of project, we compare healthy volunteers with pathological group, and we observed basically that we observed two things. First of all, that for these pathological groups, it was a hypertrophic cardiomyopathy. So this is interesting to the myocardium. The stiffness is very high. It was well known, but first time that we are able to quantify directly on our patients. And the second thing, it was a surprise for us that we observed that there was like an aging of the myocardial stiffness. If you look at the green dots, we thought that for adults, the myocardial stiffness on healthy control should be more or less the same. But surprisingly, we observed that you have a natural increasing of the myocardial stiffness, which was, for me, the most important result of this work. Other teams decide to use this second strategy, so natural waves. And so they use a mitral valve closure, aortic valve closure, atrial kick, basically to track this natural vibration and to quantify the stiffness using these natural events. But if you go a little bit more in detail, so now we'll go more to the technical part of the topic, it's not so easy. It's not so easy because when you look carefully on wave characteristics, there is two different things that it's important to integrate. The first thing is that these waves are very different. So if you look at natural waves, if you look at acoustic characteristics of this wave, and if you compare with the shear wave induced by acoustic reaction force, the frequency and the motion, so the amplitude of the waves, are completely different. And the second important information is that these waves, by definition, are either natural, so you can control the timing, or induced. So why this type of difference could impact on our capacity to quantify the myocardial stiffness? The geometry, first of all. Due to the acoustic properties of this wave, depending on their frequency and their amplitude, could be a bit forced in terms of propagation in the myocardium. So in a simple word, depending of the shape of the medium, and I will say the thickness of the medium, you could have some interaction between this wave propagation and the tissue around the myocardium. So, for example, we try to quantify that. In vitro, we try to explore some slices and to see the difference in terms of shear wave velocity depending of the thickness of the slices. And the main message of this work was to say below 4 millimeters, depending of the equipment, the frequency emission, and so on, but depending of a certain cutoff, the wave velocity is completely different in the medium. And it makes sense in terms of physics. Just the observation to say the wave is not capable to have a free propagation. So you will have some interaction with other medium. So the message is to say you can't use the shear wave velocity to assess the stiffness. It's more complicated than you could think. Other team did exactly the same in silico. And so, pretty recently, because we were curious about children myocardial stiffness and neonates, we just confirm on human that indeed, if the myocardium is less than 4 millimeters, it could be complicated to use this shear wave propagation. I'm saying that because many people here are curious on atrium. And usually I arrived a few months ago and say, is it possible to assess myocardial stiffness on atrium? And to me, it will be the main challenge. I don't have the answer yet. We'll try. But the fact that the atrium is so thin, it could be a limit for this type of techniques. Second important characteristic is the anisotropy. So the fiber orientation. I'm saying that because different teams also try to explore the fiber orientation link with the shear wave propagation. And just one slide to say that we are lucky because Sébastien Sales, which is an engineer, a PhD, will join us in 1 month now. And he's one of the experts of this 3D ultra-fast assessment for shear wave propagation. And it's something that we would like to explore here in the HU. So it is the link between fiber orientation and shear wave propagation. And so the last characteristic is the timing. So, I said you can't use every time the shear wave propagation to assess the stiffness. Indeed, for natural events, you are forced to assess the wave just after this event. And so if you use natural wave, you can control the timing. But if you use acoustic reaction force, you could push when you want and basically assess the stiffness, the myocardial stiffness variation along the cardiac cycle. And this is what we did. We just push every time during the cardiac cycle to see how the myocardial stiffness varies during the cardiac cycle. And here, just to let you know that the myocardial stiffness is not one number. It would be a mistake. Chamber stiffness could be in diastole, but the myocardial stiffness, due to the active properties of the myocardium, changes. And so the diastolic myocardial stiffness, the passive myocardial stiffness is pretty stable, but during systole, the myocardial stiffness increases because it's directly linked with the contractility. So now that we just see the way by ultrasound that we are capable to quantify the myocardial stiffness, to me, the two important next steps are these two next steps. So first of all, to link the quantitative assessment done by ultrasound with the tissue itself. So basically to link the data observed by an imaging technique with the tissue, and also to understand the active properties of the myocardium, so the loading condition on the variation of the myocardial stiffness. And it will be the two natural presentations. So Emma and Pierre are working directly on these two topics. And that's it.
Is there any question from the audience? Okay.
Great presentation, Olivia. I was wondering, what's the spatial resolution that you can have for the stiffness, and how much do you expect the stiffness to change spatially?
So our spatial resolution, I would say, it's more in millimeters. So a way to answer to this question, which is not easy. Why? Because we could detect micro displacement. So the wave, the amplitude of the wave in terms of micrometers. But our capacity to link this wave with stiffness characteristic is in terms of micrometers. And another way to answer is to say that the shear wave propagation after acoustic variation force has a huge attenuation. So the propagation will be on few millimeters and then disappear due to the attenuation. So you could really see this wave propagate in the myocardium on few millimeters and basically assess the velocities. About the variation, it's not easy to answer. Again, I think we lack of data on that. I think we could say that the shear wave velocities could vary pretty quickly on two different type of tissue in terms of millimeters. So, for example, if you have an infarct and a small one, like 2 millimeters, the shear wave velocity should be completely different in this localization, and we should be able to see it. But we lack data on that. We don't have really publication demonstrated that. But from theory, it will be the answer.
Thank you. Does this mean that when you're talking about the sensitivity and the issue that the wave is vanishing rapidly, spatially speaking? Are you talking about when you induce the wave by external stimulus, or by is this the same also from external stimulus, or is this different between the natural waves? Or
yeah, great question. It's only for external stimulus due to the wavelengths, and I could show it again. For natural wave, there is propagation on centimeters.
Does this mean that if you want to map this, you need to push at different times, at different locations, and then map at the same? Exactly.
It's exactly why I will say many teams decide to use natural waves, because in one wave, you could map the propagation. This is a 3D videos that I showed. However, you are right. If you use acoustic variation force, shear wave induced by acoustic variation force, the idea is that you will have to, if you want to map the full ventricles, you have to push everywhere. Yeah. And you can change that. It's really linked with the physics. It's also why we are talking about myocardial stiffness. I think it would be too ambitious to talk about chamber stiffness. So in a way, we say, okay, we could assess the stiffness of one segment, so myocardial segment. And to have access to the chamber stiffness, we need 3D echo, high frame rate, and tracking different waves, which is still not done. Yeah.
If there is a other question?
Olivier, can you say a few words about the new ultrasound system that you've brought to Lyric, like the advantage with regards to the conventional ones?
Yeah, it's a very simple answer. This is the only one capable to do unfocused waves in a way. Not only one, there is other one, but it's the one which is, for me and for many scientists in this topic, the more flexible one that you could control in order to generate unfocused beam. When I say unfocused, because this is by definition the ultra fast imaging. You know, conventional ultrasound, you focused your emission, and you have to repeat the focused emission in order to recreate an image. When you unfocused the beam, basically you intensificate the full medium, and so you could collect all the data to come from the medium and create an image. But in order to be able to do that, you need a hardware capable to control each piezoelectric element in your probe, and the Verasonic system is one of them. It's fully open. So the side effect, I mean the drawback of that, is that it's not clinically approvable. So that's why you need some mechanism to be able to apply that on human, but it will be cool.
No more question? I have a last one, very nice question about the time of the procedure between MRI and ultrasound, and between both, which one have a better resolution or precision?
I like the question because right now I'm working on a project and I will sell it as a echo in 1 second. So basically, the advantage of the ultrasound, so there's many advantages. And I know there's many MRI guys in the room. But it's pretty mobile. So you could move in the hospital or in your lab where you want. It's pretty compact. And the advantage of ultrafast, more specifically volumetric ultrafast, so 3D ultrafast, that theoretically, in one acquisition, like in 1 second, you could get your full data set. So see everything everywhere. And we think that now technically it's possible. So you could do a full echo in 1 second. Then there is the post-processing, which is another question. But in terms of acquisition, to answer to your question, it's extremely quick.
There is any reaction? Okay. So thanks a lot for your presentation. We will now hear Pierre. He will present the stiffness in the right ventricle in pulmonary hypertension in patients.
Okay, so hello everyone. My name is Pierre Garvinet. I am a cardiologist. I just finished my subspecialty fellowship here at the University Hospital of Bordeaux. And right now I'm doing a 6-month internship as a Master's 2 student inside the Ultrafast team. And so I am very happy to have the opportunity to present you my project today, which is called Stiffness of the Right Ventricle in Pulmonary Hypertension Patients. So first, a few words about pulmonary hypertension. What is it? So it is a pretty complicated condition with a very simple definition, as it is defined by the elevation of the mean pulmonary arterial pressure above the threshold of 20 millimeters of mercury. This elevation will be responsible for clinical signs of right ventricular failure, such as edema, palpitations, or exercise limitation. And the natural course of this disease will evolve towards a worsening of the symptoms, appearance of liver failure, kidney failure, and eventually, without any intervention, to end-stage right ventricular failure and to death. It is a pretty common condition, as different registries estimate that around 1% of the global population is affected by pulmonary hypertension. So we understand that to correctly characterize these patients and take care of them is a matter of public health. So behind this pretty easy definition hide a whole lot of different etiologies, which we usually classify in 5 groups, whether the original problem comes from the heart, the lungs, or the pulmonary vessels themselves. But as you can see in this very hard to digest table, there is a long list of different etiologies inside each group. So this all leads to a great heterogeneity of the patients that we have to take care of. And we as physicians, we do not like this heterogeneity because it might be difficult to precisely assess the patient that we have in front of us and to say what is their prognosis. So over the different years, we have developed tools to try and assess their prognosis. But again, in this hard to digest table, there is a whole lot of different markers and we use a combination of them, but it is pretty imprecise. And when we know that the 1-year mortality rate can vary from 5% up to 20%, we understand that it is very challenging when we have a PH patient in front of us to answer this question: what is their prognosis? And their prognosis relies entirely on the state of the right ventricle and how advanced the remodeling effects are. So as you know, the right ventricle has a different anatomy than the left ventricle. It is much thinner as it has a lower afterload to face, and the orientation of its fibers make it a more compliant chamber than the left ventricle. And the elevation of the pressure in PH patients will induce an increase in the afterload. And this increased afterload will set remodeling effects, such as hypertrophy of the cardiomyocytes according to Laplace law, and to try and maintain a normal outflow for as long as possible by increasing the right ventricular contractility. But also fibrosis, mostly by deposition of collagen inside the tissue. But I will not get too much in details about that because this is what Emma will talk to you about in her presentation. And this remodeling effects will all lead to right ventricular failure. And what is interesting to notice is that these remodeling effects, and especially fibrosis, will induce an increase in the myocardial stiffness. So if we could measure the myocardial stiffness of these patients, then maybe we could estimate how far the remodeling effects are and how advanced the patient is in the disease. So this is the challenge that we are facing. How can we estimate the stiffness of these patients? Olivier has told you we have few tools at our disposal, but the most validated one is by performing a heart catheterization and measure a pressure volume loop to then derive from it the end diastolic elastance. The end diastolic elastance is a very powerful stiffness marker, but to obtain it, as Olivier said, is invasive. It requires catheters that we do not have routinely, and it prolongs the procedure. So, as a matter of fact, in the real life, we almost never have this information. When we evaluate the PH patient, we do not know the stiffness of his right ventricle. And yet, some studies have decided to look into it, and I wanted to show you this one. So this is a study where a PH group underwent a heart catheterization at baseline with the measurement of the end diastolic elastance. And on the left, you can see the survival curves. So the dotted line is the line representing the patients with a low elastance, which means a low stiffness. And the full line represents the patients with a high elastance, which means a high stiffness. So you can see that there is a huge prognosis difference between these two groups, as the group that is stiffer at baseline is most severe. And it is confirmed in this analysis where you can see that there is a big difference in the elastance between the control group, between the group of PH patients that survived for more than 5 years, and the group of PH patients that was the most severe because they survived for less than 5 years. Okay, so we understand that getting this information about stiffness might be a very powerful tool to evaluate these patients. So how could we do it tomorrow? And this is where shear wave elastography comes in play. So as Olivier has told you, it's a sequence where we first use a pushing mode to send acoustic radiation force to create a shear wave inside the myocardium. And then we switch to an imaging mode to track this shear wave and to measure its velocity as it is related to the stiffness of the myocardium. And we can do that at every moment of the cardiac cycle. So this will allow us to get the stiffness during the full cardiac cycle. So this is the objective of my project, to evaluate the myocardial stiffness of the right ventricle in pulmonary hypertension patients using shear wave elastography and to compare it to healthy volunteers. So how will we do that? So this project was initiated a few years ago by the ultrafast team of the Vilma Lab in SickKids, which is an hospital in Toronto. So they recruited 40 patients. The first group is a group of 20 PH pediatric patients, and the second group is 20 age-matched healthy volunteers. So all these patients underwent at baseline a normal echocardiogram, a classical one, and an ultrafast ultrasound with shear wave elastography with the method that I just described. Most of these exams were performed by Maëlys Benet, who is another cardiologist inside the ultrafast team, who is working now in Bordeaux. And then there is a whole post-processing phase to obtain from these images the velocity of the shear wave and so the stiffness of the myocardium. The other part of this project is to collect clinical data, first at baseline and then at the latest follow-up, to try and correlate the stiffness data to clinical outcomes. So my place in this project is first to do the post-processing part to obtain the velocity from all the data that was collected during the past 2 years in Toronto, and also to collect the clinical data, which I will do during a 1-month observership inside the Vilma Lab of Toronto. So a little focus on the post-processing part. So this is a little video that I wanted to show you because it's way better than words. So using the same probe, first we send acoustic radiation force to create a shear wave, and then we switch to the imaging mode to look at the propagation of this shear wave. We will then focus our attention to the area of interest. In this case, it's the septum between the two ventricles. So again, measure the velocity and then derive from it the shear modulus. And doing so, we will draw a line, and every special point of this line will be represented thanks to a code inside a space-time analysis, where the movement will be represented by red dots. And so these red lines are the propagation of the shear wave. So what does it look like in real life? So this is an ultrasound image. This is a parasternal long axis view. You can see here is the right ventricle, and this is the left ventricle. This little red dot that you see is the location where the echographist decides to, I'm sorry, I don't see it. Okay, so this little red dot is the location where the echographist decides to send the acoustic radiation force. And then the shear wave will be created inside this area. And this is where we will use the code that was generated by Jean Barranger, who is an engineer who's worked on ultrafast for a long time, to get this shear wave. And on this shear wave, we can measure its velocity and so the stiffness of the myocardium at this precise moment of the cardiac cycle. So I wanted to show you the first result. So of course, all the post-processing part is not finished yet, so I cannot show you all the data and I cannot show you any statistical analysis, but still I wanted to show you some of the graphs because I think they are pretty interesting. So this is the evolution of the stiffness during a whole cardiac cycle for one patient. This is the cardiac cycle, the electrocardiogram, and here's the stiffness. So the first interesting part is that during the systole, there is a strong increase in the stiffness. And as Olivier has told you, some authors think that this peak, this end systolic stiffness might be a load-independent marker of contractility. But this is yet to be confirmed. And then there is a drop in the velocity during the isovolumic relaxation phase, and then the stiffness stays low during the rest of the diastole. So this was the cycle of a PH patient. And if we compare it to an healthy volunteer around the same age, well, first we can see that the pattern is globally the same with the increase during the systole and then the decrease during the diastole. But if we look closer at the mean stiffness during the diastole, for the healthy volunteer, it's more around 1.4 kilopascal, and on the PH patient, more around 2.3. So of course, this is just preliminary data. It needs to be confirmed once the whole cohort is analyzed, but these are the first results and the first signals that we get from this analysis. A little word of opening. So of course, we're going to look at the diastolic stiffness, the systolic stiffness, but some authors have suggested to look at the ratio of the systolic stiffness on the diastolic stiffness, as it might be a better reflection of the force generated by the ventricle. Kainen and her team have showed that this ratio is strongly correlated with the preload recruitable stroke work, who is a load-independent marker of contractility. And if we look at these two patients that I just showed you, well, the PH patient has a higher systolic stiffness than the healthy volunteer. But if we look at the force generated by these two patients, then we see that the force is lower in the PH patients. So again, just preliminary data, but this is surely something that we will look at. And finally, a word about the future. So as you may know, we are building here two ultrafast ultrasound machines, and hopefully one of them will be available at the hospital for clinical research. And for my part, I will be back in the hospital at the end of this year. So it will be a great opportunity to pursue this type of project and to even begin wider ones. So thank you for your attention. Thank you for the LEAP teams, of course, for their support, and to the ultrafast team back in Toronto. And I will answer any questions if you have some.
So thanks for your talk. If there is any question in the audience?
Thank you for your presentation. Just have one question, maybe I didn't get it, but for the stiffness graph, do you perform several measures on the same patient, only one?
No, we
several patients.
Yeah, no, we perform on the same patient several measures, and then we take the mean stiffness, the mean velocity, and then stiffness for each moment of the cardiac cycle.
Okay, thank you.
And I just didn't show, normally on those graphs, they are the, the error bar. So I just took it off because it's more visual display, but yeah, it's, I mean.
Thank you. Thank you very much. And between the two patients, I assume they are both at rest, patients and volunteers.
Yes.
Do they have exactly the same heart rhythm? Do they have differences that could explain, for example, that you have a change?
Well, the heart rhythm might be a bit different, but they are both at rest, so it might it's pretty much the same. The conditions in which the exam is performed are the same, so there shouldn't be any other external thing that could alter the results.
Do you think that this kind of measurement would be Also, making this under exercise, for example, could bring different information, because if you're under exercise, maybe you would see a larger difference between the two groups than at rest. Has this been done?
On exercise, to my knowledge, it hasn't been done. It might be interesting to maybe look at it, but no, to my knowledge, it hasn't been done.
It's a very simple question. I hope it's not a trap. It's not in my intention to make a trap. The slide about the survival curve, you said that the higher the elastance, the worse it is.
Yeah.
Okay, so but what is the elastance? Is it 1 over elasticity?
No, the elastance is the slope of the tangent to the end. I'm going to show you so this curve in the PV loop, we have this curve, who is the end diastolic pressure volume relationship. And when we look at the slope of the tangent to this curve at the end of diastole, well, this slope is the end diastolic elastance. So the stiffer it is, the higher the slope is. So the higher the elastance is.
Yeah, and it's bad to have a high elastance.
Well, it means that the stiffness, but right now, as Olivier told you, we are talking about chamber stiffness, which we can derive some parameters of myocardial stiffness. But yeah, it means that your stiffness is higher.
Yeah, because I have no intuition what is elastance and stiffness. I mean, materially, if you have to explain to a kid, for example.
Well,
intuitively, what
there is a great to a kid. I wouldn't know, but it's the stress response to it's the stretch response to the stress. Okay, so when you apply stress to material, how will it strain? That's the elastic properties that we're looking at. It's not clear.
If you apply a force, if it deforms a lot, it's, I don't know.
Well, that means that the elasticity is pretty great and the stiffness is pretty low.
Okay, elasticity. So elastance is 1 over elasticity. Can we say that?
No, maybe I could.
Yeah.
It's a very important question. It's basically there is confusion between the physics concept and physiological concept. So if you ask a cardiologist, the answer will be the one given by Pierre. But by definition, the elastance is the contractility. And the contractility, by definition, is the capacity to generate a stress, whatever the loading conditions are. And so the definition of contractility is not easy to understand, but this is the one. So it's a variation of stress induced by the tissue. Another way to answer is the Looks law. So the link between stress, stiffness, and strain. So the contractility directly is linked with the stiffness and strain of the medium. I will be more than happy to discuss more in detail with you. I just want to highlight the fact that these concepts are really a bridge between physics, physiology, and medicine. And usually there is a confusion between these concepts. So elastance and contractility are not the same, but they are considered the same in medicine. And maybe one thing about your question, Bruno, I just want to add on what Pierre said. The age is very important. So we age match, you ask for heart rate and so on. So we learn from the past that the age modifies the stiffness with the same hemodynamical condition. And So, I was thinking about the exercise. This is a conclusion. So we did it, and it will be published soon, I think. But we on animals we use dobutamine to modify the contractility, and so the elastance on the PV loop. But we don't have any drugs to modify the compliance, as you know. So we modify the contractility, and we observe a huge difference in terms of stiffness. And Mathieu Pernaut published in 2011 in JACC a direct link between systolic stiffness and contractility. Yeah. with the same drug.
So the question you mentioned during your talk that there is difficulty in measuring the wave propagation if your weapons correctly the size, the thickness is around 4 millimeters. What was the size? What's the size of the chamber you're measuring?
Well, of course, it depends a lot on the age of the patient and on the fact that it is a healthy volunteer or not, because the PH patients often have a right ventricular hypertrophy. But it can vary from a few millimeters from the healthy volunteers that are very young, that are some month old, to more than 5, 6, 7, 8 millimeters on others. So there's a great variation. So of course, we will take that into consideration, especially for the youngest patients, as their velocities might be a bit slower than they would be supposed, are most difficult to measure. Yeah, that's certainly one of the challenges. Yeah.
And correct me if I'm wrong, Pierre, but I think we don't have yet this data. I think it was the data that you will collect in Toronto.
It is. Yeah, Miley showed me some of the details, but yeah, we do not have the full list.
Yeah. I'm saying that because for the right ventricle, exactly like as I said for the atrium, we don't know yet. We don't know the exact thickness of this right ventricle, the free wall of the right ventricle, and how it will impact on the shear propagation.
Is there other question? Okay, so thanks. We will now hear Emma on a subject about the myocardial stiffness to the DNA, from the DNA, I think I was missing something, to the organ.
Thank you for your introduction. Hello everyone, I'm Emma Robin, and I'm going to present you also my project for this last presentation of this scientific talk. So I'm currently in my 6-month internship to validate my engineering study in biotechnology in Strasbourg, and I joined the Olivier Ultrafast team 2 months ago to work on the myocardial stiffness from DNA to the organ, and particularly about the collagen and tiling remodeling in the tetralogy of Fallot. And this project is like a fusion of 2 scientific domains, the medical imaging and the biology, and I want to try to combine them to better understand the myocardial stiffness. So as Olivier and Pierre explained to you, stiffness is a real important characteristic of the myocardium. It can be either a consequence of cardiopathies, like the pulmonary arterial hypertension, which Pierre works on, or like in tetralogy of Fallot. I will explain you a bit later this pathology. But myocardial stiffness can also be a cause of some pathologies like restrictive cardiopathy. So, like Pierre, my project will focus on myocardial stiffness as a response, as a consequence of a pathology where there is a right ventricular remodeling. So when there is this type of remodeling, you can observe physiological effects on the muscles. This observation can be done notably with the ultrafast techniques, as Olivier explained to you. This brings lots of information of these effects of this stiffness. And my N2 project is more about to bring new information on the molecular and cellular origin of this remodeling. So, as I told you at the beginning, my work focuses on collagen and tiling in the tetralogy of Fallot cardiopathy. So a bit of introduction for the tetralogy of Fallot cardiopathy is the most common cyanotic congenital heart disease, and it's characterized by pulmonary stenosis, a ventricular septal defect, dextroposition of aorta, and an hypertrophy of the right ventricular. To better understand the long-term complication after the surgical repair in the lyric, repaired tetralogy of fallow pigs models were made. So the pigs model got a three steps surgery. The first one is to create a RV volume overload. The second one is to create a RV pressure overload. And then they made a right ventricular outflow tract scar. This pig model is interesting in my project because the RV pressure and volume overload leads to a remodeling and maybe impact the myocardial stiffness. And this is what I'm going to try to understand. A bit of introduction about the collagen. This is a structural protein of the extracellular matrix of the cardiomyocytes, as you can see in the figure. I will focus my work on the type 1 and type 3 collagen. Type 1 is more about the resistance and fineness of the muscles, of the myocard, and type 3 to the elastic properties and the pumping function of the heart. Total collagen is already known as a stiffness marker when it increases, and lyric teams found similar results showing that the collagen content increase in Hertog model than controls. So here the goal is to complete the knowledge of the remodeling of the collagen with a focus on the type 1 and type 3. Now the tidine is known as the largest human protein. It's an intracellular protein localized in the sarcomere in all muscle types. And tidine works as a molecular spring responsible for the passive elasticity of muscles. You can see on this figure its precise localization from the heart to the sarcomeric area. And each cardiomyocyte co-expresses two tidine isoforms, N2B and N2BA. As they don't have the same structure, they don't have the same effects on the cardiomyocyte elasticity. So especially in the heart development, where there is a switch in the expression of the two isoforms, N2B isoform is known as stiffer than the N2BA, which has a longer change and is more compliant. So my project goals is to quantitatively assess myocardial collagen and tidine in a setting of loading condition variation. To do all this study of remodeling, I'm working with Sabine Charon for the pathophysiology department. And thanks to her, I have an access to the molecular lab and all the equipment. And she also already did some general study on the expression remodeling of the tetralogy of fallow model. So me, to characterize the remodeling of these components, the stellar components, I can do analysis at the mRNA levels, the transcript study. To do that, I can do quantitative PCR. It's a technique allowing the analysis of the mRNA of a target gene. To do qPCR, you need primers, which target the gene of interest. For the collagen, the qPCR primers were already available, so I use them. And for the tidine ones, I designed my own primers. On the figure, you can see the tiny isoform structure, and the arrows represent the zone which my primers target for the qPCR. The green one targets the N2BA isoform, the red ones the N2B, and the blue ones the N2BA and N2B. So in addition to analysis of the mRNA levels, I want to do analysis of the protein themselves. The type 1 and type 3 collagen can be quantified and analyzed by Western blots and polarized microscopy. And for the tidine isoforms, I can do analysis with SDSPage. SDSPage is a technique that allows the separation of protein according to their molecular weight, and after that you can analyze them. So all this molecular remodeling comparison will be done on Hertog model against sham, who represent here the control groups. So I can now present you some of my primary results. It's the same as Pierre. I don't have any big significant data, but I show you my first results. I already did mRNA analysis on 4 tetralogy of fallow models and 4 shams. They were both euthanized 4 months after their surgery, and I analyzed these 5 genes in the pigs. At each time, I analyze the right ventricular endocardium. My graph shows the expression of the gene of interest. Here it's the COL1, the COL type 1. The expression is relatively normalized according to housekeeping genes. For type 1 collagen, there is no significant difference between the two groups. The type 3 collagen is downregulated about 1.64 times in the fallow models than shams. For the total titin, there is no significant differences between both populations. The N2B isoform is upregulated about 1.96 times in fallow than shams, and there is no difference in the expression of the N2BA isoforms. So as I explained you just before, the N2B is stiffer than the N2BA, and here this switch of isoform expression on the Hartov model is really interesting because the Hartov model got a right ventricular pressure overload that is expected to induce a higher stiffness of the myocardium. So yeah, now a few words about perspectives. I have 4 months of internship left, so I will continue my analysis on protein with Western blot and SDS page, as I told you before. I will also observe collagen fibers under polarized microscopy, and I also want to do this remodeling analysis on volume overload model only to compare them with the tetralogy of fallow model, which has volume overload and pressure overload. And all of these experiments are the starting points of a potential future project where we could create new animal models, then scan them with ultrafast to assess their myocardial stiffness, and after that correlate this data with histological and protein and molecular analysis. And this type of project will make it possible to directly link the origin and the effect of the myocardial stiffness. So yeah, thank you for listening, and I will also thank the team at the LIRIC and at my school to make this project possible. Thank you so much.
Thanks for your talk. Any question from the audience?
Thank you for your talk. Do you know if there is an evolution of your amount of collagen type 1 and type 3 with the evolution of the disease? Do you know if it's the ratio, for instance, is different with the evolution of the disease?
In tetralogy fallow?
Yeah.
No, I don't see any publication on that. On fallow, all the data of collagen I already heard about is the total collagen and never the switch of the two types.
Because sometimes with the remodeling, especially in infarct area, the amount of collagen type 1 and type 3 is different with the age of your infarct.
Okay.
So maybe.
Okay, thank you.
Yeah, that's a very cool question because I think you will be able to answer.
Yes, I will doing it when I can observe the fibers under microscopy or with Western blot, but
because you have the samples basically.
Yes.
We have different age of evolution of the pathology, so yeah, we will try to see.
Here I use animals with 4 months with the model of fallow before teenage, but there is models with 2 months, 4 months, and 6 months. So I can observe the regulation at a different time of the pathology.
Have you observed that in other type of pathological situation? I mean, do you have experience in it or?
No, because I'm not working specifically in this kind of disease or in fact, but I'm working a lot of targeting with MRI collagen type 1 and collagen type 3. And I saw that there is a different amount in the cardiac tissue, in the conductive fibers, and also it evolves with age. And sometimes it could be it could be a good marker for the evolution of some disease, for the evolution of some remodeling, and that's all. But it's just the beginning of what I observed, what I read on the literature. So yeah.
So by MRI, you can separate collagen 1 and collagen 3?
Alors, you have to validate it by histology all the time, but we can perform some molecular imaging, and we have different contrasts with magnetization transfer imaging with the collagen of type 1 and collagen of type 3. Yeah. But we have to validate with histology. Yeah.
Super cool.
Other question? I have another one about molecular biology. Which type of housekeeping gene did you use from normalized audio data on collagen and titin?
I use GUS-B and HPRT1. Those are the genes used by Sabine for the cardiac tissue. Globally, when you want to use housekeeping genes in other type of molecular qPCR, you can use myosin, things like that. But in the cardiac cells, myosin does not have the same expression if there is pathology or aging. So yeah, this is GUS1 and HTRP1.
Okay, thanks.
Sorry.
I think that's all for now. Thanks a lot for all your presentations, and go to drink.